Well, hello again, everybody. My name is Arif Kasim. I'm a science coach with Frontier School Division. I'm here joined with me, Lee and Susan. We are doing Envirothon training, and they will be discussing the topic of aquatics. So Lee and Susan, thank you for coming, and we wish to hear more from you here.
All right. So we're going to switch to the PowerPoint right to begin, and we're going to start talking about water and aquatic ecosystems. Good to go? Okay, so for you guys that are the chair people for aquatics in Envirothon, just know that this is an enormous topic, that you're covering an entire ecosystem, everything that lives in it, the physical processes, all of the different components of aquatics. So there's a lot to cover. And for this reason, I really encourage you to go and watch the previous live stream videos. We've tried to cover different things each year in live stream. Because there's just too much to cover all at one time. So we have 2 previous videos from the last 2 years, and those are linked on the Envirothon website. So those are, in my opinion, sort of the best studying that you can do. Read your reference materials, do all your practicing, but definitely make sure you watch all of the videos. Today, we're really going to focus on the physical and chemical sort of aspects of aquatic systems, looking at some cycles, water and nutrient cycles. We're not going to talk much at all about sort of things that live in aquatic systems or other pieces like that. So this is sort of more focused today. So to jump in, we're going to talk about sort of the main cycle that you need to know as an aquatic person is the water cycle. So this, obviously water links sort of every part of the terrestrial and aquatic ecosystems, atmosphere, and so we'll just walk through those different phases. Water has obviously gas, liquid, and solid phases, and all are represented in the environment. So we're just going to pick a spot. We'll start here with precipitation. So this is water in the form of liquid or frozen that's falling from the sky onto the landscape, and it falls both onto terrestrial environments and aquatic. And from here, it can stay as surface water, so things like lakes and rivers, even puddles, those all count. They're water exposed to the atmosphere. Or it can percolate through the ground, through cracks or through the soil, and end up as groundwater. And this is either sort of in the soil, water that's going to be taken up by plants, or as sort of aquifers and larger bodies of water under the ground. From here, once the plants take up water, then sometimes they lose some of that. This is basically like the plant sweating. It's called transpiration, and that emits water vapor back into the atmosphere. And that type of water going back into the atmosphere also happens through evaporation from different water bodies, oceans, lakes, rivers, puddles, everything. And from there, you either just have really humid air where lots of water in the atmosphere, or when you start to see, you get really sort of dense amounts of water in the atmosphere. You start to see things condense into clouds, and then from there, of course, you get precipitation over again. So make sure you know the water cycle. When we're looking at water quality, this is basically water is a universal solvent, or quote, a universal solvent, meaning that things are very easily dissolved in water, and it dissolves almost everything. Well, not almost everything, but many things. So there are many dissolved substances present in water, even if it just looks like clear water. And almost everything that we as humans do affects water quality. Any water body that we interact with is going to be affected by our actions. Activities that we do, such as adding substances, we can alter sort of pathways of water with water diversions and dams. We can remove water, like for a drinking water supply, or add water, say in a sewage outfall. Lots of different activities affect water, not to mention sort of recreational water uses like boating and swimming. And the impacts, of course, are hugely varied, but we do a lot of things like increase nutrients in water, we alter the pH, we can increase pollutants and other toxins in the waterways, we can increase turbidity. There's sort of an endless list of human impacts on water. So when we want to know what sort of water quality looks like in a water body, we're going to do a chemical test. And in this way, we take some of the water some of the water out of the water body. That's the target, and we're going to test that. We can do a bunch of different types of tests to try to figure out what's in there. And the important thing when we're doing a change on the landscape is that we test the water before we make that change, and we test the water after. So when we're sampling for chemical tests, we're going to want to obviously first collect the water sample. And there's a good video on Think Trees TV that you can see a method of collecting a water sample where they use a Van Dorn water sampler, and we're actually going to demonstrate that in a second. There's also a method where you use a pump and tubing. So I have a video of that I'm just going to play. So you're going to lower the tubing of your sampling device down to a particular depth in the water column. And from there, you've got a pump that's drawing water up from the source. So the source being the depth of that tubing. This is actually me sampling water. And so you can see that I'm going to rinse the bottle a couple of times before actually collecting the sample. Because you want to just make sure that there's nothing residual in the bottle, even if it's been washed out before, that's going to alter the chemistry of the water that you're looking at. And you can see also from this video that people sample water at all times of year. So it's not just people sampling water when the lakes and rivers are open, but sampling on the ice actually is pretty easy because you can just walk out to wherever you need to get your sample from. So that's one method. And then the other methods, we're just going to demonstrate right now. So I think if we can switch to the other video here, we're going to show you the Van Dorn water sampler, and then just a dip method. Yeah, the dip. It's over there. We good? So this is if we ask you to collect a water sample on Envirothon with the Van Dorn sampler, this is what you're going to get. There's lots of different types, and basically it's a tube, hollow tube, you can see through there, that has an elastic on the inside, and these balls sort of plug the ends when they're triggered. So you lower this into the water, and then once it's triggered, it will just maintain that bit of water that was in the tube at the time that you triggered it. So you can go to whatever depth you want and get water from that depth. So for this one, you just have to pull the elastic and pull the put these little pieces, they're little metal pieces on the ends, and you have to thread this other metal piece through. So you're just basically capturing the two ends on a stick so that when you lower it in the water, so if you're going to lower it into the water to the depth, it's going to fill with water, and then you're going to trigger it from the surface by just jogging it up and down. And in doing that, you can see that the things close, and you pull it up to the surface, and then you've got a little spigot here where if you just release the seal, the water is going to start flowing out of the tube here. So we might ask you to do that. The other method Susan's going to show you. This is the simplest of simple methods for collecting water.
Technical. This is a stick with a peanut butter jar on the end, made by one of the Envirothon volunteers for us. So this is a simple way to just lean over and grab a sample of water from the surface. Well, you could go a little bit deeper, but basically it just gives you, sorry, could you hear that? Yeah, it's on. Yeah, it's on. Can you hear me? Okay, so you basically just lean over and take a dip out of the water. So with this highly technical piece of equipment that you might get to use at the Envirothon.
Yeah, so that would be called a grab sample, and there's lots of grab samples collected in aquatic ecology. Okay, we can go back to the PowerPoint. So after you've collected a water sample, you might want to separate the dissolved stuff from the particulates that are in the water, and so you might filter your water sample. And we did a pretty thorough demonstration of this on the 2017 live stream video, so definitely go take a look at that. And your basic idea is that you're going to pour the water through a piece of material, whether it's a filter made of paper or glass or whatever, and you're going to end up with any particulates that were in the sample stuck to the filter, and anything that's dissolved will have gone through the filter and be collected in the water sample. Sometimes when you're looking at the chemistry of the water, it's really important for you to know the water temperature. Water obviously moderates temperature, so in summer a water body is going to be cooler than the air. In the winter the water body is going to be warmer than the air. And aquatic organisms can really only tolerate a certain range of temperatures. Water temperatures also signal to organisms when to lay their eggs, when to migrate, all sorts of things. So the temperatures of water bodies is really important. Factors that affect water temperature include the size and depth of the water body, the color of the water, and the turbidity, or the amount of particulates. Whether the water is sort of static or flowing, so in a lentic system like a lake where there's not a lot of turbulence, or a lotic or flowing system like in a river, you'd have different temperatures. And of course, the season of the year and the latitude of the water body will affect temperature. We talked a lot about the stratification of water bodies, particularly lakes, in the 2017 live stream video, looking at the different layers that develop in water bodies with temperature. So we're not going to talk about that today, but definitely go and take a look at that. And there's also a good video on the Think Trees TV YouTube channel of measuring water temperature. So if you go to the Manitoba Forestry Association YouTube, you can see an aquatics video for measuring the temperature of water. So we're going to do 2 demonstrations of methods that we might use at Envirothon, or ask you to use, to measure temperature in the water. So we're going to switch over here in a second. But just to start, I'm going to just show one picture because I don't think the thermometer we have will show up very well in close-up. But this is just a basic thermometer where the bulb has the liquid in it, and the liquid will expand or contract depending on the temperature it's submerged in. And you can see here, you're going to measure in increments of 1 degree. So the temperature here is 19 degrees. You've got just 1 tick below 20. And we don't, if the measurement is sort of halfway between a degree, just round up or down wherever it's closer to. You don't need to be that specific, just every degree. Okay, so we're going to switch to actually measuring the temperature. So we've got one example here. Susan, do you wanna do this
no.
with this one?
No, go ahead.
Okay, so we've got 2 examples. We've got this digital thermometer with a little probe on it, which we might ask you to use. And to do that, you're just gonna stick the probe into the water sample and let the temperature stabilize and see. So you can give it a second. And we've also got just a little aquarium thermometer in here that's gonna that you're just going to read and tell you what the water temperature is. So you probably can't see it on there. This says it's just under 20 degrees. And the digital thermometer here says 20.1. So pretty close. That's as close as we're gonna ask you to be. So these are 2 methods that we might ask you to read on Envirothon. Now it says 20.0. Oh, sorry. All right. Where did I put the thing? Ah. Okay, we can go back to the PowerPoint. So when you're doing a chemical test, there are some safety considerations to make. That in a field or in a lab situation, you want to make sure you're wearing the appropriate personal protective equipment, or PPE. And this will really depend on the type of chemicals you're handling. Sometimes if it's nothing very toxic, you don't need anything, or you don't need much. And if it's a more harmful substance, then you need to take more precautions. For an Envirothon test, the stuff that you're working with, you only ever will have to wear gloves and goggles. There's nothing really toxic that we're going to ask you to handle. But of course, in an analytical lab setting, things like lab coats and face shields, maybe a hazardous materials suit, fume hood, all those types of things are possibilities, depending on the nature of the chemicals you're handling. Important that you take care when you're handling your samples in an Envirothon event. There's lots of activity, lots happening. You don't want to spill your sample. You don't want to spill your sample on someone else. So just make sure that you're careful when you're doing the tests. And just practice really good sort of clean technique in terms of your gloves, that you don't want to have stuff on your hands and then touch doorknobs or touch other people or whatever. That just, if you keep things to just touching your sample and your equipment, and just avoid sort of touching anything else when you're doing your samples. You want to just do your test and then get it done. Okay, so we're going to start with a test that actually doesn't require any personal protective equipment, and this is a salinity test. So Mitch was talking about salinity in the soil presentation, and now we're going to talk about salinity in water. So this, of course, being the salt content in the water. So fresh water, lots of lakes and rivers in Canada are fresh water, and these have basically no salinity. There's basically no salt in them, really low concentrations. You get brackish waters where ocean meets fresh water in estuaries and stuff, and in some other situations. So that's sort of the range of higher salinity values. Once you get into the ocean, of course, it's about 30 to 50 parts per trillion of salt, and that's that really, if you taste the water, it's quite salty. And then you get the really high salinity, greater than 50, in brine pools, things that are just like salt, super, super salty areas that not much is going to be able to live in, maybe some really specialized organisms. Saline water is denser than fresh water, so if you do have a freshwater system that has some salt content, often what happens is you get a layer of saline water right at the bottom, and then the fresh water is flowing over top, and not a lot of mixing between the two. And organisms are generally adapted to the salt content of their home environment, so you're not going to be able to take an organism from a freshwater environment and just toss it into the ocean and it's going to be fine. There's a lot to do with sort of water passing across their membranes that they're adapted to in their specific environment. Some organisms do go between saltwater and freshwater, but they have sort of specialized structures or physiology in order to be able to do that. And so, of course, a change in salt content of an aquatic ecosystem can really change that ecosystem and the organisms that live in it. For example, if you have road salt running off into a freshwater ecosystem, changing the salinity, or if you have injections of saline water used in hydraulic fracturing or fracking, those can all change those aquatic ecosystems. Okay, so we're gonna do a salinity test using a piece of equipment called a hydrometer. So this is just measuring a change in, or the density essentially of the water, and then we're gonna convert that measurement into salinity using a conversion table. So because these instruments are pretty small to read, like the temperature, I'm also gonna show a couple of pictures first. So this is the stem of the hydrometer that has the different increments on it, and you sort of have to twirl it to read the whole thing. So I've got two pictures. And you can see at the top that we've got 1.000. And then as we go down, 1.005, 1.010 essentially. And the part that you're going to have to pay attention to are the tick marks that I've highlighted in yellow. So they're the 0.001 increments. They're not the tiny little ones on the right, but the sort of bigger tick marks on the left. And so, for example, if you have your hydrometer sitting in the water and it's measuring like it does what it looks like here, that water is touching the hydrometer at 1.001, so just one tick mark down from the top. So that would be your hydrometer measurement. Another example here, just a video of a hydrometer sitting at 1.10. So you can see it's 10 tick marks down from the top. So that's your hydrometer reading there. Okay, so we're going to switch over to this camera here and look at the salinity measurement. So we've got two different samples here. And so this is the hydrometer. It's just a basically weighted end. It's going to bob up and down, and then there's the different increments on the side there. I don't know if you can see. Doesn't matter. And we have to, it's important to measure temperature when we measure salinity. So we've got a thermometer in here, and we're just going to let that settle And we've got a second one here with a different salinity measurement. Do you want to, yeah, do you want to talk about that? Yeah.
Oh, I just wanted to point out that the hydrometer, when you're using it, it's glass.
Yeah.
And it's quite fragile. So don't be too rough with it.
You shouldn't really have to touch it.
But
If you have to spin it, you're going to want to spin it a little bit and then wait until it settles. Mostly you just want to be able to sort of move around and read it from there. So this is the table that I was talking about in terms of reading the salinity measurement. So what you observe on the hydrometer is down the side, and the temperature of your water is across the top. So when you look at the observed salinity and the observed reading and the temperature, it's going to tell you what the salinity of your water is. And this actually, I think that one doesn't go high enough because it's warm in here.
What did you want to do with this guy?
Yeah, do you want to show them just where you're reading it?
Okay. Can you see this?
Sort of.
Step back. All right. So do you see how there's a slight curve? It's called the meniscus. You want to read, if you can see it, you want to read at the bottom, not up here where it clings to the surface of the graduated cylinder, because water does that. You want to read it on the flat part. So you would take the reading there.
Yeah, so you're basically reading it right where the water touches the hydrometer. It's maybe going to go up slightly here, but it's going to be so small that it's not going to matter. So don't look from the side of the cylinder. Actually look where the water touches the hydrometer. Okay, so let's take the readings here. Maybe Susan, do you want to look at the temperature for each in comparison? So the temperature reading on here is, let's see, 19. And the hydrometer reading for the first one, it's just one tick down, so like in our picture, so 1.001. So if we look at the table, what does that tell us?
The table?
Yeah.
Okay.
Can we see?
The reading would be 1.9 at the intersection of temperature across the top and your reading down the left, and 1.9 is what you would get.
And then the other one, it's more saline. There's some salt in here. And so we're 16.5 ticks down. We're going to round up and say 17 ticks down. So 1.017.
And is it the same temperature?
It's the same temperature.
So now the salinity reading would be 23. Can you see that?
So we've got the first sample in the freshwater sort of range. We're a little bit on the salty side maybe for freshwater, but still basically fresh. And then we're verging on ocean. This is brackish to ocean in the second sample. Okay, so you might have to do that on a test. Okay, so we'll go back to the PowerPoint. The next test we're going to talk about is turbidity. So turbidity is a measure of how cloudy water appears. And the focus is on the amount of particulates in the water, not on the color of the water. So a low turbidity sample could still be dark, but completely clear. So think about how those two are different. Clear water may appear cleaner than turbid water. You might think, oh, this really muddy water is much dirtier than clear water, but clear doesn't necessarily equal healthy. You can still have sort of pollutants or toxins in very clear looking water. Sources of turbidity to water bodies include stirred up sediment. If the water's flowing, it really mixes everything together. Runoff or erosion from streams going into a water body. Plankton, if it's high algal content in the water, you can have a lot of little particulates. And the effects of turbidity on aquatic systems means that it prevents the transmission of light sort of through the water. So it will slow photosynthesis. There'll be less organisms sort of in the deeper depths able to photosynthesize. And it can raise the water temperature. Those particulates are going to heat up with the sun and then impart that heat on the water body. So we're just going to show you a couple of demonstrations. I'm sorry to make you switch back to the other camera. So this, we'll use a hydrometer again. This would be the example of just clear water, not turbid, not dark. So we've got just really clear and no particulates floating in there. And then next we've got this water that's not dark, but it's cloudy. So you've got this real sort of, you can't see through it very well. It's it's turbid water. Another example, you can compare this to the clear sample. So this water is pretty dark, but you can see through it still. I don't know how easy you can see it, but it's still pretty clear, not a lot of particulates or cloudiness there, compared to a sample that's both dark and turbid. So you've got particulates in here, and it's really, you can't really see through this water, whereas this one is much more clear. So just a comparison of those two, and then the combination of those two. So just think about the difference between the sort of darkness of the water and the particulates in there. The other thing is when you filter a sample, of course, you're removing the particulates. So a sample of water that didn't have particulates in it, you might end up with a filter that looks like this. It looks like there's basically nothing on it, even though stuff has been filtered through. Whereas in a system where you've got more turbidity, you're going to end up with a filter that's got a lot of particulates on it. So these are both taken from the Red River, and we filtered 50 milliliters of water through this one and 100 milliliters of water through this one. So you can see this one's got basically twice as much particulates. So that would be from a sample possibly that's just got the high turbidity, possibly it was the dark and turbid sample. But these ones are these are taking all the particulates out of the water. Okay. Okay. So we're going to move on to nutrients and start looking at some of the cycles. And the first one we're going to talk about is phosphorus. So phosphorus is essential for life, for metabolism and growth of organisms. It's really important in energy. And if you're looking at the chemistry, the phosphorus ion plus 4 oxygen atoms equals phosphate. So this is what we're going to actually test for. And orthophosphate is the free form, so it's just dissolved in the water, in the environment. It's not bound up in plant or animal tissue. So the test we're actually doing is an orthophosphate test. Freshwater systems, phosphorus is generally the limiting nutrient. So that's where you end up with these different designations of eutrophic, mesotrophic, oligotrophic systems, with eutrophic being lots of nutrients, mesotrophic being sort of a moderate amount of nutrients, and oligotrophic being very low amounts of nutrients. And in those types of systems, you sort of accordingly get similar amounts of growth of algae and other organisms. So eutrophic systems will have a lot more growth and support more life than oligotrophic systems. Sources of phosphorus to aquatic environments include fertilizers, detergents, although there is a phosphate ban on many types of detergent because of the impacts they have on fresh water, certain industrial wastes, and of course human and animal wastes as well. Impacts of excess phosphorus getting into aquatic environments leads to eutrophication, like we talked about, which means that algae just sort of go out of control in the lakes and they bloom. So you get these big, dense accumulations of algae called an algal bloom. And of course, after the algae live their life in these blooms, they die, they sink to the bottom, and they decompose, which it can use up basically all of the oxygen in the water body. And this low oxygen in the entire water body can lead to fish kills. So these are instances where many individuals from populations of fish end up dying simply because there's not enough oxygen. So that's obviously an enormous ecosystem impact. And the other thing is lots of the algae that really thrive in high phosphorus situations produce toxins, neurotoxins, hepatotoxins that can be really lethal to humans and to other organisms. So there's lots of impacts that affect us and the ecosystem. So we're going to demonstrate the phosphorus test that we might ask you to do on an Envirothon test. Okay, I'll get you to. So first, Susan's going to put on her gloves and her goggles.
Oh, I should have checked the glasses first. This could be your biggest...
So this is the type of test that we're going to have you do in any situation. We have a number of different kits like this, and they're all basically the same type of thing. So you get a card that has instructions on it that will tell you in the steps what you're going to do with the equipment provided. And then usually on the other side, there's some sort of color chart, because what we're looking for is a color change for this test. So once you've put on your gloves and your goggles, you're going to look at the equipment that you're provided, and you're going to read the instructions here. So I'm going to read them, and Susan's going to do the test. So first, you're going to fill the test tube to the 5 milliliter line. So each test tube will have different lines on it. You won't be able to read them on the camera. There's 5 mils, there's 10 mils, and the different tests will tell you what volume of water to use. So you want to be as close as you can to that. Don't worry about being perfectly bang on, but as close as you can to that amount. And then you're going to add 1 phosphorus test tablet. So these little tablets, just like a blister pack, and you're just going to pop one of those little things into the tube. And the easiest way is to sort of hold it over top. Just let the little thing drop into the tube.
This guy broke.
Got a broken one. Just put the piece in. It's fine.
Yes. I'm just checking. Yep. Okay. Got it.
So you're going to cap the thing, and you're going to shake that tube until the tablet has disintegrated. So maybe I will shake that one if you want to get the other one going. Oh.
And it fairly aggressively.
Yeah, shake it fairly aggressively because they take a little while to dissolve. Okay. So Susan's going to do, we've got 2 samples here, and we're going to test the 2 different samples to see if they're different. And then the important thing on the card is that you wait 5 minutes. So we'll provide you with a timer, and you don't want to check your sample until that timer is complete. And you're going to start your timer once the whole tablet is dissolved. So you want to mix it until that is dissolved, and then you can start your timer, and then you can read it after. If you read it in the middle of your test, then you might get a result that's not accurate. Okay. So we'll just shake these. Keep shaking them until they're done. I can start talking about the phosphorus cycle. Sure. While we're shaking them.
I can shake for you if you want to.
It's all right. I'll go with it.
Tell me when you're ready to start the timer.
Oh yeah, mine's good.
Okay, I'll start yours, and then mine will be shortly after.
Okay. So on a test, we have these nutrient cycles for you in your resource materials. They may not be exactly what we show you here, but all sort of similar information. And these things, we're not going to ask you to spit them out verbatim. We'll probably provide you with the cycle and ask you for some information based on what you can see on there or your knowledge of the nutrient. We're not going to ask you to just draw out a cycle. And obviously, the important things for aquatics are things related to aquatics on the diagrams, but these include the interactions with the terrestrial environment. So the inflows of that nutrient into the aquatic system and how that system contributes that nutrient to the terrestrial environment is all important. So if we start here looking at how phosphorus ends up in waterways, it can run off after being accumulated from a variety of sources like fertilizer, like phosphate extraction from mines, like erosion from phosphate rocks, and through organic decomposition of animals and plants. So from there, that stuff ends up in the aquatic ecosystem, usually in the dissolved form. So the phosphorus has dissolved in water and has flown into the water. From here, it can be used by the aquatic food web. So it can be taken up by plants, be taken up by algae and other sort of plant-like organisms. And from there, it can move its way up the food web. So into the zooplankton, phytoplankton, into the benthic invertebrates, into the fish. And from there, those guys will die eventually. It will go into the things that eat them. It might just fall to the bottom and be incorporated into the sediments and sort of end up in that cycling. If it's in the sediments, maybe one day it will be rock. So lots of different ways that it sort of moves through the environment. So just think about the connections. Think about how phosphorus might move through the ecosystem. Just a couple of examples of sort of the oligotrophic versus eutrophic systems. So you can often see a difference between a system that's got high nutrients versus low. And so you can see the oligotrophic ones here, the water looks quite blue and clear, and the eutrophic systems are pretty green. You can actually see the algae from space. The picture of Lake Winnipeg on the right is a satellite photo, and you can see those green algae blooms just throughout the lake. the filters presented there show on either side the sort of, you filter oligotrophic water, you don't get a lot of that algae clogging the filter, whereas if you're filtering the eutrophic system, you can get quite dense amounts of algae on there. So knowing the different trophic statuses of water bodies is important. Okay, how are we on the test?
We're only at 3 minutes.
3 minutes, okay. I'm going to go on to talk about carbon while the phosphorus test completes. So there's a couple of types of carbon that we're talking about in aquatic systems. We're not going to focus very much on inorganic carbon, which is usually carbon dioxide. This is used in aquatic systems for photosynthesis and is produced through respiration of organisms. And there's also the gas exchange between the atmosphere and the water that gets carbon dioxide into the aquatic system as well. What we're mostly going to focus on is dissolved organic carbon. So this is organic material that's dissolved in the water, and the source is usually through decomposition of plants and animals. And this is what gives water its sort of brown color. So if you make a cup of tea, the leaves that made up your tea have contributed dissolved organic carbon to your water, turning it the darker color. DOC in the water supports the growth of microorganisms, and of course it limits the transition of light through the water, because the darker the water, the harder time the light is going to have getting to those deeper depths. So just a note, I guess, too, as DOC sits in a water body, it can bleach, and this is photo bleaching, that it loses color due to sunlight. So water that has sat sort of say in a lake that doesn't have much inflow or outflow might have a really high DOC content but low color. But on the whole, darker, browner water has higher DOC than clearer water. You can do a DOC chemical test using an instrument in the lab, but it's not really possible in the field. So we tend to use a surrogate measurement, which is a Seki disc or a light sensor. Okay, 1 second. Which gives us the sort of an idea of how much light is getting through the water. This is also obviously affected by turbidity, but it gives you a good example of DOC. So we're going to switch back just for a second and look at our phosphorus test results. So you can see on the first sample of water, so what you're going to do for each card is hold your vial against the card and compare the color you see to the color chart here. So this one, we're seeing a white color of the sample, and we're seeing 0. So we would say that this is 0, and this is distilled water, so it should have no phosphorus in it. And then this next one, this is our second sample. We're going to compare the color here, and now there's a gradient and it's sometimes a little bit hard to tell. I would say that this is not as light as number 1 and not as dark as number 4. To me, this looks like a 2. So on the test, I would put that this is 2 for phosphate.
And you can go halfway in between.
You can go halfway if you think it's there. We'll usually have some wiggle room. But most importantly, you're wanting to be looking at the color against the card and comparing to that chart there.
And
not to the side, on the card. Yeah. Okay, we'll go back to the PowerPoint. So back to the testing the Seki disc measurement, there is a video on ThinkTree's TV of this. We did a live stream demonstration as well a couple of years ago of Seki. And what we're going to do now, I'm just going to show you some images. So for the Seki disc measurement, you can see at the surface, it's pretty, it's just very visible. As you go deeper into the water, it becomes less and less visible, and at the deepest point, you basically can't see it anymore. So you're taking your measurement just when that Seki disc disappears from sight. So when you pull that up, you're going to check the measurement of the line, and then see what your Seki disc measurement was, and you can translate that into light transmission information, but we would give you that formula. Okay, and I guess the one other thing here is, you remember the dark water we showed you before, that dark sort of tea stain color is what we're meaning when we're talking about the sort of DOC. The carbon cycle I'm not going to talk about a lot. I just wanted to mention it as something that you should be familiar with in terms of that exchange of CO2 with the atmosphere and the water, and also the cycling of the DOC within the water body. Mostly thinking about what the inputs are and how those affect things. You don't have to know any of these numbers. It's just mostly about being familiar and being able to read the chart. Okay, the last nutrient we're going to talk about is the nitrogen, and we're going to start by demoing the nitrogen test because it'll take a few minutes. So we're just going to switch to the other camera and do that first, and then it can wait while we're going. So the nitrate test is, as you can see, these are pretty similar, phosphate and nitrate tests. It's going to be basically the same procedure, except that there are 2 tablets for this one. So this is why it's important to read the instructions, and you have to add them in particular order. So one says nitro 1, one says nitro 2. You're going to be adding them in that order to make sure that the test goes properly. So pay attention to the directions. So this one also says fill the tube to the 5 milliliter line. So we've got half a tube of water, and it says add 1 nitrate test tablet. And then we're going to cap it and mix until that one's disintegrated. And you can do the second one as well. I'll mix that one. My glove.
You can't add number 2 until number 1.
You can't add number 2 until number 1 is dissolved.
You should have had a musical intro.
Yeah. Yeah, this one seems to break down pretty quick actually, so some of the tablets are a little bit faster. Okay, so that's good for the first one, so I'll take over on there. And Susan's going to add the second tablet to that sample. Make sure it's tablet number 2.
Yes, it is.
Make sure the tablet actually goes in. That's important.
Yes, that's what I'm looking for. I can't find it. Where did it? Okay, I'll try a different one. Oh, there it is. Oh, it was stuck to the. So this is, yeah, something to. Getting the tablets out is one of the challenges.
This one's not quite dissolved, so maybe I'll give that to you to shake,
Sure.
and I will work on this one, and then I'm going to just go back to the presentation. So we'll do the timer for this one and figure out when it's done. Okay, so in terms of nitrogen, so nitrogen is abundant in nature. 79% of the air is nitrogen, your bodies, all sorts of stuff. There's nitrogen everywhere. And it's essential for plant growth, including algae, and this is why it's considered a nutrient. Nitrogen is pretty abundant in freshwater ecosystems, and some algae even fix nitrogen from the atmosphere, meaning they can bring it in. But it's somewhat limiting in the ocean, so that's the difference between freshwater and ocean food webs. Nitrogen is also limiting in many terrestrial ecosystems, and this is why you see the use of fertilizers and crop rotation and other strategies to ensure that there's enough nitrogen in the soil when people are trying to grow things. Some plants like legumes can fix nitrogen, but on the whole it's limiting in terrestrial environments. Sources of nitrogen to aquatic environments include fertilizers, sewage, animal excrement, so things like livestock, geese and other birds can provide a lot of nitrogen to wetlands and ponds and stuff that they land on. And there are lots of forms of nitrogenous products, so nitrate, ammonia. What we're doing here is the nitrate test. This one is good to go.
Did it have the second tablet in it?
Yeah. Okay, and we're getting to the end. The nitrogen cycle, again, is a cycle that we want you guys to be familiar with, and again, particularly the aquatic component. So looking at the sources of nitrogen to the aquatic environment, things like runoff and the fixing the nitrogen by the algae. when it's in there, it can cycle through that food web. So you can enter in the phytoplankton, get up to zooplankton, fish, end up decomposing. And the different nitrogenous products are changing form sort of over time, but we're not going to ask you to memorize all of that. So just think about how things cycle and how those sources end up in the aquatic ecosystem. So for nitrogen, we're going to
you can cheat.
Yeah, so we're basically at the end, but instead of just waiting for our test to complete, we're going to read them now. So we wouldn't want you to do this on the test. You have to wait the full 5 minutes. But in this case, we'll show you the 2 results here. Let's do this one first. Yeah, go ahead. So our first sample, again, you're looking at the color between in this space here. So we're looking at maybe like a very, very, very slight pinkish hue, but probably not. So we're looking at a 0. And then our second sample, that's it. It's like off the charts. That's a serious 40. So if it's even darker than the scale goes, you're obviously going with the highest number. You're not guessing beyond there. But this is definitely not in between. This is that super dark color. Yeah. Okay, one more slide. So there are a couple of other tests we might ask you to do on an exam. These include dissolved oxygen, which you can see an example of in the live stream 2017 video, and pH or acidity, looking at the live stream 2016 video. We'll give you examples of both those tests. Both are fair game for Envirothon tests. And that's it. I guess, good luck to everyone who's doing regionals and provincials tests this year. And yeah, keep studying.
Thank you so much. That was fantastic. I don't know if you noticed, but the nitrate test matches your outfit, and I'm going to assume that was on purpose.
Definitely on purpose.
Lee, Susan, I would like to thank you. A huge thanks for coming out today, as well as to our entire lineup of experts that we had today. Arif, great job throughout the day as well. Chris, thank you so much.
Yeah, thank you.
And to all of you, if you're in the Northern Regional, we'll see you next week. And all other regions are the week after. So good luck, have lots of fun, and we'll be seeing you again. Thank you.